I'm just preparing my microcentrifuge tubes. No worries, I can do everything one-handed. I'm just preparing my microcentrifuge tubes with the initial stock solution for diluting. The first tube will get an entire milliliter of my stock solution, which is distilled water mixed with a surfactant. The surfactant is called Tween 20, but you can also use a single drop of like Dawn dish soap per gallon, and shake it up and then make your little jar. You can sort of see in the jar that it's got like lots of bubbles that form on the surface, and that's all the surfactant does, is it creates these bubbles and it stops things from sticking together. So we will have a solution that when we put our spores into, it will help stop them from clumping and sort of separate them.
Can I just ask a question?
Sure.
Does any surfactant work?
Most surfactants work. I prefer Dawn dish soap just because it's not going to have any effect on the spores, and Tween 20 as well. They're just neutral in pH. They don't really change the property of the water, and they're not going to affect the germination of the spores. So yeah, like anything that is just non-scented, that's a neutral pH. So yeah, Dawn dish soap in your water will help. I have a couple bottles of Tween 20, and I've had them for like 6 years, and I've never run out of it. It's a very efficient thing to if you get it to use. 1 second. Okay, so. I'm just going to do a little introduction. Hopefully, I'm just going to do a little introduction. So my name is Max. I go by Wumbo Myco or Wumbo Genetics. I have been cultivating for about 10 years, since I was about 18 or 19 years old. And within the last 6 or 7 years, I got really interested in breeding. Initially, there was very little information available online about breeding mushrooms. Most of what was available was on forums like the Shroomery, and the main person I saw talking about breeding mushrooms was a man named Roger Rabbit. A lot of people who have been active on the Shroomery around that time are familiar with Roger Rabbit. He's sort of a legendary figure. Hamilton Morris has even interviewed him. And the story of breeding mushrooms back then is one of a lot of, I guess, mystery. As I was diving into it, the only information I could find on how to breed mushrooms was by Roger Rabbit saying that he had used, or that people had successfully used, snake venom to sort of break down the cell wall of the mushroom mycelium and pair two mycelia together. This is not a great way to do it. Personally, just because it's hard to get sterile rattlesnake venom, and it's a dangerous thing to work with. And so serial dilutions is the most basic form of breeding. This is something that I was taught in order to do microbiological testing, so food safety testing, at a big mushroom growing facility in Sebastopol. And essentially what I would do was I would dilute our supplement powders into from an innumerable amount of grains of the powder to a less dense amount. The idea is if you have bacteria in your powder and you dilute it a bunch, then you put that on a plate, you'll get only a few colonies of the contaminant growing on this nutrient plate. This nutrient plate has a grid, and since the colonies will be a little bit spaced out, you can then count on the grid how many colonies there are, and then quantify. You can tell how contaminated your food is. Okay, it will have the same percentage of contaminants as before you diluted it, but they'll be far enough apart to where I would be able to just say, okay, well, for each square there's 3 colonies of bacteria or yeast or mold, right? And that would be able to, then I would be able to say, okay, well then if there's 3, it's 300 parts per million or something mold. And that is within or without, outside of or within the safe limits. And there's, protocols that tell you what the safe limits are and stuff. So from there, I started looking into serial dilutions, and I found how to dilute spores. And that's the beginning of how I learned how to breed mushrooms in a safe and easy, albeit tedious, way. Breeding mushrooms with serial dilutions is not the most fun thing in the world because it requires a lot of pipetting and a lot of plating. After you get a small enough dilution of these spores, it will take a lot of work to verify that you have single spores, that only one spore germinated and did not pair with another spore. And I'm getting a little ahead of myself, but the idea is that from here, I wanted to find out other methods. And I have a Patreon, Wumbo Myco at patreon.com. It is on there. I have methods on how to do your serial dilutions and take that, and instead of doing 2, use 1 and pair it with already grown out mycelium of another strain, as well as a sort of a novel method that I don't like saying I created it because it is information that already existed, but at the same time is many different research papers that I sort of pieced together to I guess, create this method of pairing plate mycelium together of 2 strains to recombine their genetics. So I'm just gonna start the slide here to give us something to talk about. I have basically already discussed this. So it's very difficult to count individual spores when they're in high concentrations. And so by diluting them, you can take the innumerable amount of spores and make them something you can count. For example, 1/10 of a milliliter of your original sample is going to be added to 9/10 of dilutant, which is your sterile water. And the dilution factor is 1 to 10. If you keep doing that, the next one will be 1 to 100, then 1 to 1,000, and 1 to 10,000, and et cetera, right? But by the time you get to 1 to 1,000, even if you have so many spores, thousands of spores in your original solution, you should be able to reasonably assume that a drop of this solution will have about 1 to 5 spores in it. So you can drop that onto a plate and let it germinate and be reasonably sure that they're not going to germinate and touch each other and then form an already mated pair. The reason for this is that we want unmated pairs to mate with each other intentionally, right? So this is sort of a diagram on how the dilution works. It'll show you, based on density, that the liquid is getting a lot more clear, right? A lighter color. And it clearly shows you, this is more with like blood, that there is very few platelets in the 10 to the 6th power than there is in the 1 to 10. That is the whole concept between serial dilutions. It's very simple. And so basically what I'm going to show you is how to prepare the original spore suspension. So this is your first tube. What I do is I get these microcentrifuge tubes. They're 2 milliliters, just so I have a little bit of headroom to shake things up and all that. They are made of polypropylene 5, that I can take them and put them into this tube rack and put that in my pressure cooker and sterilize it. So that I know, or sorry, this is my pipette tips, duh. I can put them onto this tube rack and put that in my pressure cooker, and I can steam sterilize it and know that what's in the tubes is totally sterile, and that I don't have to worry about any other things germinating on the plate when I put them on there. So what you would do, basically, is you would take your pipette tips, your tubes, and your sterile or your distilled water with your surfactant in it. You put all that in the pressure cooker, sterilize it. Now you have it on your workstation, and we're going to do the first thing, which is putting our spores into 1 milliliter of your distilled water with your Dawn dish soap, basically. So, maybe I'll just put it here. So, on a spore print, generally, if you can see the spores, you can be pretty sure that there are too many spores to count. So what I do is I take a nice sterile scalpel, and I just scrape the spores very gently. And a lot of times what they'll do is they'll stick to your scalpel. So then what I'm going to do is I'm going to put them into the top of this and just tap them in there. And when I shake it up, you'll be able to see that, I mean, it's hard to see, but what I like to do is I like to keep going until you can really see some spores in there. So I might even scrape some more in there. Yeah, so for scraping, what I like to do is I keep the blade sort of angled so that I'm not cutting into the foil at all. I'm just scraping along the foil. And now again, I have more spores, and I'm going to stick it in and remove them from the blade, put them into the tube. Obviously, if you have two hands and all that, it'll be a lot cleaner of a process. And so you mix it up, and then if you do end up looking at this a little closer, you would be able to see that the water has turned a bit purple because we are working with locyb cubensis, which has a purple-brown spore print. Now that you have your first dilution rate, all your spores suspended in 1 milliliter of water, you're just going to take a new clean pipette tip, and you're going to attach it to your pipette. You don't need a mechanical pipette. You can use little pipettes that are, 1 milliliter and ones that are 0.1 milliliter, whatever you need. But these mechanical pipettes are awesome. The tips are reusable. I just wash them and then sterilize them. And yeah. So what I'm going to do is I'm going to turn my mechanical pipette's top until the numbers start going down. And that is going to set the rate of volume, or the volume of liquid that it's going to pull. So if you're looking for one of these, you're going to look for one that says 100 to 1000 UL. The U is a funny little U that means microliter. And on it, when I was getting 1 milliliter, I'm doing 1000 microliters. That's 1 milliliter. And now to get a tenth of a milliliter, I'm going to set it to 100 microliters. So it takes a little second because it's a lot of turning. It does it by 10 every time, 5 actually. 100 microliters, and when I push the plunger down and then pull it very gently up, you can see this 1 milliliter pipette is not quite full. It's basically only a tenth of the way full. So what I'm going to do is I'm going to put that. Now, I did get a little ahead of myself, and I forgot to prep the rest of these, but you're going to put the tenth of the milliliter into this container here, the first dilution step. And then I'm going to reset and put 0.9 milliliters, or 900 microliters, into each of these tubes. So now I'm going to take 1/10 of a milliliter from this tube. Well, let me show you first. You can just shake it like this, or what I have is it's for making like lip gloss or makeup, but it's called a vortexer, and they have fancy lab ones for labs and all that. But if you plug it in and you press down, it'll just spin and create a vortex in this tube that'll mix everything for you real well. So now I'm going to take 1/10 of a milliliter from this tube, and I'm going to put it in this tube. Such a basic process that it is almost annoying to describe. Yeah, it's a little boring, but you got to start somewhere with breeding. So you make sure it's really evenly distributed, all the spores. Shaking it like this also helps activate the surfactant to knock all of the spores away from each other so they're not stuck, because they're so small that they will use sort of a static or whatever force that applies to them will sort of cause them to want to clump together. And now 1/10 of a milliliter into this tube. So what I'm going to show you is basically a tube that is, getting stuck to my fingers. Tube that is your stock, your stock solution, which is nice and purple. You can visually see the spores in it, which means you're not going to be able to count them. This is your 1 to 100 dilution, your 1 to 1,000 dilution, and your 1 to 10,000 dilution. Right? So what happens now is that these two, your 1 to 1,000 and your 1 to 10,000, should have somewhere around the right amount of spores that you're looking for. What I like to do when I want to be sure is I will take from the 1 to 1,000, you just take a little bit, and I have some microscope slides on the table here, which are just these thin pieces of glass, which you put under a microscope. And I put a single drop from my 1 to 1,000 on the microscope slide. And then what you're going to need is a cover slip. This cover slip is just a little piece of even thinner glass that when you put it over the drop of water, it is going to sort of seal down and make it so that you have a lens when you put it under a microscope. I was hoping to have a microscope today, but things happen, and what you're going to do is just take this, put it under a microscope, and look at it, and if you see a lot of spores, you know you have not enough dilution, right? So what I look for is, I look at it under a pretty low magnification, and I see, can I count a bunch of little dots here? Is it more than 10? Is it more than 5, 10, something like that? And it's probably too much. So, yeah.
So I noticed you went all the way to a 10,000 dilution.
Mm-hmm. Yeah, because what I like to do is I like to look at the 1,000 first to make sure that if I select the 10,000, it's not too few, right? If I end up selecting the 10,000 and I can't see anything, then you could do it backwards, right? You could select the 10,000 first, and if there is no spores that you can visualize, then you go back to the 1,000. But I like to choose the 1,000 first, see how dense it is, and then go to the 10,000, and yeah. So now, we have all these dilutions, and this is your way of verifying, okay, I'm at the right dilution rate, which you don't have to do at all. Like, you don't have to do this step at all if you don't want to. You can just plate both of them. But now you have a microscope slide. You're going to take that, put it under your microscope, look at it under probably 100 times magnification or something like this, and then maybe even 400 times magnification if you want to see them up close, and just see generally how many spores are in there. If there's more than 5 or 10 spores on that slide, then you have too many spores. So you're going to want to do another dilution. So there is no limit on how diluted you can make the an original spore solution. The idea is you want to work with what you can visualize, and honestly, when I'm looking at the 10,000, I can't see a single spore with my naked eye, and that's a good thing, because spores are not really very visible to the naked eye. So, at least not when they're alone, right? When they're clumped up together, that's when you can see them. So, now we have our dilution of our first strain. I only have this one spore print with me today, but what you would do is this exact same process, which I don't need to show you again, but you do this exact same process with your other strain and go and visualize it. And now that you have both of them, you're going to take a new pipette tip. And, oh man, you know what? I don't have Petri dishes, but, you know, let's pretend you got a Petri dish here. You're going to take a Petri dish that has agar in it. And a lot of people just call them agar plates. You can buy them already sterile if you don't know how to pour them or want to or have a clean space to do it. And you're going to plate just a single tiny little drop on that plate. What I like to do is I take a plate spreader, which is just like a piece of metal, which is flat, and I will just spread it around the plate and then seal it up and let it germinate for about 1 week to 30 days. After 1 week to 30 days, you will see 1 to 5 little dots on the plate. At first, what they're going to look like is little scratches. So let me go through here, and you might be able to get a better visualization about what I'm talking about. So this is all the subsequent dilutions, final dilution, and counting. Here's what, under about 400 magnification, what a spore is going to look like. Spore is, I believe, a golden brown spore, so they might be a little bit of a different color. But like here, under 100 times magnification, these are your spores of your locyb cubensis that are typical. This is a purple-brown spore, and you can see here, these ones, even with a surfactant, want to stick together. They're clumped up, and that is very hard to separate. So that's why we look at it under a microscope a lot of times, is to see, like, okay, do we have a lot of clumping? Do we have too many spores? This, I would probably use an even further dilution with this instead of this dilution. This is probably like 100 or 1,000 times diluted. Yeah, and then after that, we're going to pair the monokaryons. So a lot of times this is not monokaryons, but this is a spore germination. But what you're going to see is a lot more like up in the top left corner of this agar plate, there is sort of a very scratchy, hard to see, very light white mycelium growing there. Monokaryons usually don't look rhizomorphic, which is sort of this ropey, thick growth, and they usually don't look like this dense, fluffy growth that has sort of a aerial quality to it where it's coming off of the plate. When spores pair, that's when they do this. They have this, but there is no one look for a monokaryon, so you don't want to just go off of what it looks like on the plate. I'm just giving you a reference for what you're going to see on that plate when they germinate. So yeah, what you need to do is you need to take ones that are not touching any other ones. So you find a little germination on there, a little scratchy white puff coming off, and you're going to take your scalpel and you're going to transfer that to a new Petri dish and let that grow. That is isolating a monokaryon. A monokaryon is just a germinated spore, a single spore that contains half of the chromosomes it needs to create a mushroom, right? Mushrooms are really interesting in that they contain 2 nuclei in each cell, and they need those 2 nuclei in each cell in order to actually create a mushroom. Otherwise, they'll just grow out, keep eating and eating until they find that pair in which they can combine to become a dikaryon, meaning that it has 2 nuclei. So after you select your monokaryons of each of your strain, you isolate them to individual plates. You can now take transfers of them and plate them on the same plate. So you're going to do this systematically. And to do this systematically, it's going to require you to pair, let's say, Hanoi and Avalanche. These are two strains that I have that are pretty prevalent in the community. I will label each monokaryon as Hanoi 1, H1, and H2. Let's say I have two monokaryons. And Avalanche as Avalanche 1 and Avalanche 2, A1, A2. This is the permutations you can have between them. You can pair H1 with A1, H1 with A2, H2 with A1, H2 with A2, right? These are all the possible ways you could pair them. And so you are going to do that on individual plates. And this is generally how you're going to pair them. You're going to put the individual monokaryon here and there. I like to do 2 on a plate just because it saves plates. And usually if you put them far enough apart, they're going to touch each other before they touch on the other side. Now, this one right here, these are already dikaryons. This is a project that I was doing that was not mono-mono pairing. This is di-mono pairing, which is also possible, but it is harder to confirm the success rate of. So when you have 2 monos and you want to pair them, what can you do to tell that your single spore germinations have actually mated with each other? Well, you look at the mycelium under a microscope, and it's very easy. You would take your mycelium on a petri dish, you would cut out a little wedge, and you would place it on your slide, and then you put a cover slip over it, and it will not fall down all the way, right? There's agar underneath, it's 3-dimensional. So what I do is I take a little pencil and I lightly tap, trying not to break the cover slip, to crush the agar, the nutrient media, underneath the mycelium, and that actually acts as like water under the slip where it seals it down and makes it a nice little lens. So this is called a crush mount or a smash mount, squash mount, whatever you want to call it. But basically you're putting the mycelium under the slide, putting the cover slip on, crushing it down, and now you can look at the mycelium under the microscope. So what are we looking for? We're looking for a clamp connection. A clamp connection is a trait of mycelium. It's a structure of mycelium that it will grow after it pairs. Now, this is not true for every mushroom, but when we're talking about locyb cubensis, this is always true. Or at least almost always true. I'm sure with science there's always exceptions, right? But in every instance I've ever seen in my life, a single spore that germinates will have no clamp connections. And spores that have then been, single spores that have then been paired together will have a clamp connection. The clamp connection is a structure that regulates the amount of organelles in a cell. So if your cell has, it needs a nuclei, it needs a vacuole or some sort of organelle that's in the cell that has a function, it can basically act as a barrier so that they don't leave, or as something that allows extra organelles to pass through what's called a septa. It's hard to see, but there's like a light line right at the end of that clamp connection. That's called a septa, and it is just a barrier that stops things from moving between the cells. So all we're doing is we're looking, usually under 400 times magnification, to find a clamp connection to say, yes, this is a dikaryon. This is paired, it's mated, this was a successful mating, right? After you have a successful mating, we're there, right? Now you have a dikaryon, you're going to put it to grain. You're going to put it to grain, you're going to grow it out, and then you're going to use that grown mycelium to put into substrate and grow your mushrooms. The first pairing that you have of a mushroom that's newly mated is going to have a lot of variation possible in it, because these 2 nuclei have just now combined half of the genome of each strain and created a new genome that has, like I said, half of each parent. Now, how they express in physical traits is going to be very different. than just half of this one, half of this one. So the next step is phenohunting. Phenohunting is also sort of a complex process. It can be very much a pain in the ass, but what you're going to do is you're going to grow those mushrooms out, you're going to look at what they look like, and then you're going to select for the traits that you want. When we breed, a lot of times we're selecting for physical traits. I'm selecting a lot of times for novelty. Does it have a cool stem, a thick, dense stem? Does it have an interesting cap, color, or spore color? All these kinds of things, right? You're going to take the fruits that you like the best out of your tub or your bag or whatever it grows in, and then you're going to take spores from that. And we're going to repeat the process all over again. Although we don't have to do the pairings. We're going to do phenohunting. So you're just going to put those spores willy-nilly onto a plate, let them germinate, select, and you're going to be a little more selective in how you allow that plate to grow again. So when you germinate those spores from the first pairing, it's going to have all sorts of different types of growth. And so what you're going to do is you're going to take your scalpel and you're going to cut into maybe like 5 or 6, maybe even 10. The more you do, the better, because you will get the more chances of selecting every possible variation that arises. And you're going to put those on individual petri dishes, let them grow out until they show only one form of growth. And then you're going to do the same thing. Send it back to grain, fruit it out. Look for the same trait you selected for, take spores from that. Each time you germinate those spores, you're going to the next filial generation. So I like to call it F1 when I have my first pairing and I grow it out and I take those spores. And then I germinate it, and that's F2. And then I go all the way to F3, and now you've actually isolated the spores. So the variation available in the spores is going to be cut down by about 90%. After you've isolated the spores by filial selection, you're then going to start cloning. I like to take clones of each generation just so that if I really need to and things start growing whack and I can't find the phenotypes I really liked, I could just go back to that clone, which is almost guaranteed to grow like the mushrooms that I liked before and selected from. So I will go through 3 filial generations and then 3 clone generations, and then I have a fully isolated strain. It's now pretty much definitely going to grow the way you want it to with the traits you've been selecting for. Now, if you're more interested in chemotype, if you like the potency of the mushroom that you've been selecting for, you're going to have to, at every step, send it in for testing, come back, say, okay, this is the amount of psilocybin, psilocin, baeocystin, norbaeocystin, aruginacin, all the different compounds that can be in a psilocybin mushroom. And then you're going to take the clones or the spores from the ones you like the most with and do the same process, right? Now, I don't technically select for psilocybin very often, because a lot of times what happens is people get the inclination to want to isolate the most potent mushroom possible. I personally don't think that's a good idea. With cannabis, what we see is that people look for the stronger and stronger and stronger strains with THC. And what that led to in the cannabis industry is less and less and less CBG, CBD, all the other cannabinoids that are present in it, which actually augment the THC. THC is a very anxiety-heavy molecule. It can sort of be very racy and unenjoyable for a lot of people when it's pure. And when you add all the other cannabinoids and terpenes into that, it mellows it out and it gives you a more full spectrum experience, which is where a lot of the medicinal benefits come from. And so I feel the same way with mushrooms. I think that even though we don't have a lot of actual studies showing you exactly what these alkaloids do, like baeocystin or norbaeocystin or norpsilocin, all these things, we don't really know what they do inside of you. I believe that they're an important part of the whole experience of eating a mushroom, right? And so I like to select for ones that have, if I am looking for chemotype, interesting, novel compounds that aren't psilocybin inside of it. Higher baeocystin levels, higher norpsilocin levels. But other than that, I generally stay away from that kind of thinking and just look for the ones that look cool, that have good growth patterns, that have resistance to bacteria or contamination, all of this kind of stuff. For the people who just came in, I'll give an extremely brief rundown of spore isolation, which is what we've been talking about. Serial dilution is the process of taking spores, putting them into a stock solution, and then trying to take what is innumerable, uncountable by the naked eye or any reasonable means, and make it countable. Because what we want to do when we are breeding mushrooms is take one single spore, which has half of the genetic material needed to make a mushroom, and pair it with another single spore, which has the other half of the genetic material needed to make a mushroom. So this would be two different strains or species or whatever you're trying to breed together. A lot of times, different species will have barriers of incompatibility between them. So most of the time when we're doing this, we're talking about the same species, but just different strains. So that process was basically me taking these tubes, putting 1 milliliter in the first tube of sterile distilled water, mixed with a surfactant, which is mine is tween 20, but it could also be Dawn dish soap. And a surfactant is just something that it foams up, and it makes it so that things that want to stick together sort of want to separate from each other. The goal here is to make it so that your spores are not stuck together, and they're not going to, when they get onto a nutrient media like an agar plate, they're not going to germinate right next to each other and then pair immediately, right? You want to get them unpaired. So the process of serial dilution is taking 1 milliliter of sterile water, filling it with spores, then taking 1/10 of a milliliter of that spore solution and putting it into 9/10 of a milliliter of sterile distilled stock solution, and then doing that again and again and again until you can be pretty sure that every drop is just going to have one spore on it. 1 to 5, but generally 1 is what you're looking for. So now that you've isolated single spores, you've paired them together, you've confirmed that they have no clamp connections, because when you have monokaryons, they need a clamp connection in order to, or rather, when you have monokaryons that pair and mate with each other, they form a clamp connection. So you're making sure that your single spore isolates have no clamp connections, and then when you pair them together, you're making sure that they do have clamp connections. This is a way to confirm that it's a successful cross. Now, this is an excerpt from a paper that I really like. It's one that I have on my Patreon where I go over. It's modeling the consequences of the dikaryotic life cycle of mushrooms, mushroom-forming fungi on genomic conflict. So when you breed mushrooms with different methods, it will change the way that the populations of genes in that mushroom act. So you have, generally, in every mushroom, populations of genes which want to affect the vegetative fitness, which is how the mushroom grows on media, how fast it grows, how fast it can consume nutrients. You will have reproductive fitness, which determines the rate at which the mycelium produces spores. And then you will also have, oh man, it's cut off. You also have mating fitness, which is generally how willing a mycelium is to donate or receive a nuclei from another organism. And that last part pertains more to other breeding methods. But generally, when we are breeding with this mono-mono, we call it the diploid scenario, which is just a single spore meeting a single spore and making a mating pair. You will see all these dots go from the corner that shows vegetative fitness and mating fitness, and over about 100 generations of these pairings will focus towards spore fitness. So what it's isolating for is the most productive spore-generating mycelium. And other methods of breeding, like dimono, pairing a single spore with an already formed dikaryon, that will sort of keep the populations very even. And di-di, pairing two already formed dikaryons together, will destroy the spore fitness and select for mating fitness. So we can use these different breeding methods in order to affect the gene populations in an intentional way. So what I like to do is I start with die-die mating because that is the more detrimental to the spore fitness and is going to make it become sterile and stop producing spores if I continue to do this type of mating. And instead of continuing this type of mating over the generations, I stop it, take the successful result, and start doing these mono-mono pairings to produce better spores, basically, to make it so that you can keep breeding the mushroom forever. So yeah, that is, it's a very basic overview of breeding mushrooms. Overall, I think that anybody can do this, and anybody can understand this. All you have to do is memorize a couple of stupidly complex words, unnecessarily complex words from microbiology. But other than that, it's conceptually very simple. Yeah, and that's it. I should have included some pictures of some of my successful pairings and the mushrooms they produce, but if you come up to me, I'll show you. I have some stickers here of Avalanche and Khmer Kong, which are some of my more popular strains that I've produced. And yeah, thank you so much. If you have any questions, please feel free.